Good day everyone, I'm Deepa Manivasadan and today we the members of Group 11 will be presenting about Hepatitis A virus for our oral presentation.
Hello everyone, my name is Kagirsi Morgan.
I'm Ragini K.
Hi, and I am Ranjita Devi Saravanan.
Hepatitis A virus or HIV is a remarkable virus with unique characteristics that make it resilient and adaptable in various environments. Let's dive into its structure first. HIV is a small virus with just 27 to 32 nanometers, belonging to Picornaviridae family. Its size and non-enveloped structure make it extremely resilient. Unlike enveloped viruses, HIV can resist drying, detergent, and harsh environments. This means it can survive in food and water, making it a significant concern for foodborne illnesses. Structurally, it's an icosahedral virus, which is symmetrical with 20 triangular faces. Its genome is single-stranded RNA, specifically positive sense RNA, allowing it to directly serve as an mRNA for protein synthesis. The virus has structural proteins that form its capsid, which are responsible for its stability in the environment. HIV's capsid protein, like BP1, provides stability and enable interaction with host liver cells, crucial for initiating infection. This interaction is a key step in its ability to infect humans. Now that we understand its structure, let's explore its remarkable environmental adaptability. Since HIV is non-enveloped, it can survive in water, soil, and food for long periods. It resists stomach acid, even many common disinfectants, and freezing temperatures, which may contribute to its persistence in food items such as frozen or refrigerated produce. In addition, HIV is highly durable and able to survive in various environments for extended periods. In fresh water, it can persist for 10 to 100 days and in salt water for several weeks. On surface like plastic and metal, it lasts days to weeks, making it persistent. Shellfish like cockles pose high risk since HIV can survive freezing and remain infectious. Contamination from polluted waters compound this issue. Fortunately, HIV can be inactivated by heating food to above 85 Celsius for 1 minute or using proper water chlorination. The main mode of transmission is the fecal-oral route is typically spread through contaminated food or water, often linked to poor hygiene, like not washing hands after restroom use, leading to contaminated food or water. In summary, HIV is resilient in harsh environment and its ability to spread through contaminated food and water make it critical pathogen in food safety.
Hello everyone, I'm going to talk about the key characteristics of Hepatitis A. Here are some key characteristics. Firstly, transmission. Hepatitis A spreads through the fecal-oral route. The virus is transmitted when the food or water is contaminated with the stool of an infected person. The poor hygiene practices such as unwashed hands or unsanitary food handling are common causes of outbreaks. Secondly, symptoms. Infected individuals may experience fever, fatigue, nausea, abdominal pain, and jaundice, which causes yellowing of the skin and eyes. Lastly, high-risk foods. The virus is often linked to foods that are not cooked after contamination. Raw or minimally processed foods handled by food preparers pose a significant risk. Relevance to the suspected food items. All the food items served at the festival were contaminated with foodborne pathogen, which is Hepatitis A, but the overall risk level is low. However, 3 specific food items were highly contaminated and their risk level is high. The first food is kerang rebus or boiled cockles. The risk level is high. Shellfish like cockles can be contaminated with Hepatitis A if harvested from contaminated water. Even if boiled, there is no guarantee that all the virus will be killed. Since Hepatitis A is waterborne, contaminated water from which the cockles were harvested could introduce the virus into the food, especially if water treatment practices were inadequate. The next suspected food item is chendol, a dessert with shaved ice, coconut milk, pandan jelly, and palm sugar syrup. The risk level is high. Chendol involves multiple ingredients, some of which could be vulnerable to contamination. The shaved ice and raw coconut milk used in chendol are susceptible to contamination through improper handling or from contaminated water sources. Hepatitis A virus can survive in cold, moist conditions, and poor hygiene during the preparation or serving of this dessert could lead to contamination. Ice, in particular, is often handled without sufficient cleanliness, making it a high-risk item for Hepatitis A virus transmission. The last suspected food item is fresh-cut fruits. The risk level is high. Fruits can be contaminated by Hepatitis A virus if they are not washed properly or if contaminated water is used during preparation. Even though fruits are typically not cooked, they are still susceptible to contamination if they are handled by unclean hands or exposed to contaminated surfaces or utensils. Hepatitis A virus can remain on raw produce, especially if it is not properly rinsed or sanitized before being served. Additionally, fruits served without cooking present a higher risk compared to cooked items.
Next, I'll talk about the analysis of the possible sources of contamination, the three key areas which are improper food handling, cross-contamination, and environmental factors. First, let's look into improper food handling, which plays a significant role in the spread of hepatitis A. When food handlers fail to wash their hands properly after using the toilet, fecal oral transmission of pathogen occurs. Example is the hepatitis A virus. Chendol and fresh cut fruits are high-risk items because they are often require no further cooking to eliminate pathogens. Using bare hands to handle ready-to-eat foods increases the risk of contamination. Gloves or utensils should be used as a barrier. Under cooking, high-risk foods such as boiled cockles, which is Kerang rebus, can allow the hepatitis A virus to survive. Proper cooking at high temperatures is essential. Inadequately cleaned cutting boards, knives, and equipment can spread the virus from one food item to another. Next is the cross-contamination, which occurs when pathogens are transferred between food surfaces or utensils. For example, fresh cut fruits can be contaminated if the same cutting boards or knives are used for fresh cut fruits and cockles without proper cleaning. Storing raw and cooked foods together without separation allows pathogens to spread too. Proper storage practices with clear labeling and separation are crucial. In buffet-style settings, shared utensils and overcrowding can facilitate contamination. Cross-contamination often occurs when utensils used for raw shellfish such as Kerang rebus are not properly cleaned before being used for other foods. Finally, let's talk about environmental factors that contribute to contamination. For example, ice or coconut milk is used in chendol may be prepared with untreated or dirty water. Boiled cockles sourced from polluted coastal areas are a natural reservoir for the hepatitis A virus. Leaving foods unrefrigerated in warm conditions creates an environment where pathogens can multiply. Last but not least is the common sources or reservoirs. Raw shellfish, like cockles, naturally carry the hepatitis A virus when ousted from polluted waters. Contaminated water supplies, whether for drinking or food preparation, are another frequent vector. Food handlers also can spread the virus to others. The three most high-risk foods associated with hepatitis A outbreaks are cockles, chendol, and fresh-cut fruits. The most two high-risk food will be cockles and chendol. In conclusion, the sources of contamination, whether through improper food handling, cross-contamination, or environmental factors, can be addressed with strict hygiene, proper cooking, clean water, and storage practices. By identifying and mitigating these risks, we can significantly reduce the spread of hepatitis A. That's all from me. I'll pass to my friend.
I'll present about ISO protocol for hepatitis A virus detection in food. Our main reference material is the ISO 15216-1 guideline. There are two forms of sample that we'll be looking into. One is solid sample, which is boiled cockles, and another one is liquid sample, which is chendol. The first step is sample collection and preparation. For solid sample, 25 grams of boiled cockles in a sterile container is collected and then blended with 225 ml of sterile phosphate buffered saline (PBS), which contains 3% of beef extract. For liquid sample, 50 ml of chendol is collected in a sterile container and it is mixed thoroughly to obtain homogeneous solution. Second step is pre-enrichment. The samples are then combined with an extraction solution like beef extract, which has a pH of approximately 7.2. Then the mixture is agitated for 30 minutes at 4 degrees Celsius to release the hepatitis A virus particles. Then it is centrifuged at 10,000 relative centrifugal force for 30 minutes to concentrate the viral particles into a pellet. The third step is selective enrichment. The viral pellet is then eluted by resuspending it in 1 times PBS, and then the eluate is filtered using a 0.45 mm membrane filter to remove debris. The fourth step is nucleic acid extraction. Extract hepatitis A virus RNA from the concentrated sample using commercial RNA extraction kits like Yagen viral RNA kit or other kits available like Ribopure or Trizol. The fifth step is detection by reverse transcription qPCR. Here we are following ISO 15216-1 guidelines, so we have to use HAV specific primers and probes targeting the 5' non-coding region of hepatitis A virus. A reaction mix known as RT-PCR, master mix, and thermal cycle program is prepared as per the kit specification. Sixth step is confirmation and biochemical test. For confirmation, gel electrophoresis is done. Run the RT-PCR product on an agarose gel to confirm the presence of the expected fragment size. Expected amplicon sizes typically ranges from 70 to 150 base pairs depending on the primers used. For sequencing, sequence the PCR product to verify its identity by matching it against reference sequences in database like GenBank. Here's an example of agarose gel electrophoresis showing RT-PCR amplicon. approximately 250 base pairs of dark viral hepatitis A virus. This is an example. For biochemical tests, the ISO method itself does not include traditional biochemical tests since these are unsuitable for viruses like HAV, which cannot grow on artificial medium. However, immunoassays like ELISA can be used as complementary tests to confirm the presence of viral proteins or to investigate outbreaks serologically, though they are not mandatory in the standard. Otherwise, we can also use fecal contamination indicators, for example, coliform testing, which might indirectly support findings but are not part of the hepatitis A virus specific ISO test. ELISA is a serological test done to detect antibodies. The seventh step is serological and molecular confirmation. For molecular testing, firstly, RNA is extracted, then RT-qPCR is done, and then the CT value is interpreted. CT value is the cycle threshold value. Hepatitis A virus is detected if the sample produces a CT value less than or equals to 40. Lower CT values indicate higher levels of hepatitis A virus RNA. For serological testing, enzyme-linked immunosorbent assay ELISA is done. It detects HAV capsid proteins, antigens, or specific antibodies in samples. It also produces color change results that correlate with viral presence. Here, the picture shows the comparison between instant ELISA assay and conventional ELISA assay. The eighth step is identification and reporting. Use RT-qPCR results to confirm the presence of HAV RNA in the food sample. Determine the amount of HAV RNA based on CT cycle threshold values. Lower CT indicates more virus. Include food sample tested like gram-repose and chendol and results of testing in the report. Here's an example of the result table that can be included in the report. This table consists of the items, the gram-repose and chendol, the results, whether it's positive or negative and the reason behind it, the process control which shows the CT value, and the interpretation which shows whether there has been contaminated occurred or not. In a nutshell, we as a part of team of food specialists, we believe that by identifying the morphological features of hepatitis A virus, how HAV could contaminate the food that was present in the food festival, identifying the sources of contamination, and by designing an ISO testing protocol, we will be able to identify the relevance of hepatitis A virus to the pathogen outbreak.